audible and audible mics are participants.
Hello, participants. Is it audible to you?
Yes, sir, you're audible.
Okay. Shall we start the program now? Okay. Esteemed Secretary Madam of our college, respected Principal, the organizers Dr. Binukumari and Dr. Tenmuli, and the distinguished guest of the program today from Singapore, dear participants and other persons, good afternoon to everybody. It is a very happy occasion that in the lockdown period we are organizing a webinar on the basic science research, a tool for nation building. So most essential topic, almost nowadays throughout the world, all the science institutions like colleges and universities, they are doing actually the basic research, the outcome of which definitely most useful for innovating the new technological tools. So application point of view, the basic science research is most needed at this juncture. So the basic or the fundamental science research is about the knowledge of environment and then the nature, the physical and chemical forces, and the major goal of the basic research is to understand the function of the materials newly discovered. So in order to know everything and also to sensitize the young researchers and the faculty members, this webinar is conducted in this lockdown period. I think definitely this will be more useful for all researchers to do research in new areas in course of time. So I welcome you all for this webinar, and formally I call upon Dr. Binukumari to welcome the participants. Now it will hand over to Binukumari. Madam.
A very good afternoon to one and all present here. I am Dr. S. Binukumari, HOD in charge, Department of Zoology, Konganadu Arts and Science College. I am extremely honored to have the opportunity to address in this prestigious moment. It gives a great pleasure to welcome all participants attending this webinar on Research in Basic Science, a tool for nation builder, jointly organized by Research and Development Cell and Dr.M.Aruchami Research Foundation. I am privileged to welcome our Honorable Secretary and Director of our college, Dr. C.A. Vasigi Madam, who is the source of inspiration and support in all our accomplishments. Welcome you, Madam. I am pleased to welcome our dynamic principal, Dr. M. Lakshmanasamy sir, who is the backbone of all our endeavors. Welcome you, sir. I am happy to welcome Dr. Palsamy sir, Dean R&D, Dr. S.R. Madan Sankar, Dean Academics, Head PG Research Department of Biotechnology to this webinar. Welcome you, sir. A hearty welcome to today's resource person, Dr. Parasuraman Batmadaman, Deputy Director, Ling-Kong Chain School of Medicine, Nanyang Technological University, Singapore, who is going to deliver his thoughts on I view the topic, role of multimodal imaging in drug discovery. Welcome you, sir. I extend my warm welcome to all faculty members, scholars and students of Konganadu Arts and Science College and participants from other institutions for their active participation in this webinar session. Welcome you all. Thank you.
Now I invite our Secretary Madam to inaugurate the function. Before inviting her, a brief introduction about our Secretary. Basically, she is an academician and working in the Konganadu Arts and Science College as Assistant Professor of Zoology in the early 1980s. After then, she had an experience in working with the Borkovic Laboratory, UK, and then she is a research fellow in the PL 480 program also in 1980s. She was the council member, Indian Science Congress Association, a biggest scientific forum in the world. And now she is the convener, Indian Science Congress Association Coimbatore chapter. So she is a highly enthusiastic. Her enthusiasm is highly contagious and very much involved in academic activities and render a great assistance both financially and morally to do always the research work. So in addition to the regular teaching and other extension activities, he is giving more importance to research activities also. So such a very good personality we are having as secretary. Now I invite our secretary madam to inaugurate the program. Now our madam.
Good afternoon to everybody. I'm very happy to join in this webinar, which is on research in basic science and tool for nation building. Thank you for the organizers, Dr. Paul Swami, who has taken a lot of effort to organize such a program. And also let me thank the Dr. Parasuraman Padmanabhan, who has consented to give a very nice topic. What he has selected is role of multimodal imaging in drug discovery. So today it's going to be a very enthusiastic and very informative session, and I'm glad and I'm participating in it. Before telling about starting this program, I would like to give a brief to the participants about our institution. Our institution has completed nearly 48 years and of service. And when this institution was started, it was generally any education institution, when they start give preference for arts courses like English literature, commerce, where the only classrooms are needed. But whereas our institution, when they started, it started with science courses. Our college is still, like, we have the strongest departments are our science departments, all basic science for that matter, from botany to zoology, physics to chemistry. And now we have bloomed in all the applied sciences like biotechnology, biochemistry, computer science, commerce, English literature, so on and so forth. So that is the brief history about our college, how it was started, and the reason you may think why science is given so much importance, because this institution was started by a scientist. So that's one of the reasons. Still, we have that tempo in, that's my father, Dr.M.Aruchami, who's the foundation is on. And he actually insisted that basic science should, on any accord, should not die, and it is the contributor for the various branches that has developed recently after 80s only. When you see after 1980s, only various other subjects like biotechnology, biophysics, bioinformatics, and microbiology, and so on and so forth, so many areas have diversified. Before that, it was only these four basic science subjects where were really the contributors for all the majority of the inventions, I would say. And even recently, like, a very of the plastics, as you know, had been one of the very major uh, um, pollutants towards our atmosphere. And thanks to the basic, our scientist, Dr. R. Vasudevan, who is from Madurai, Tyagaraja College. He is called the plastic man. He has devised a technique where these plastics can be used in laying rubberized roads. So how it can be recycled. So this is one of the contribution for which he was given the Padma Shri award by our government. And again, as this topic says, it's for a tool for nation building. Yes, of course, India, at the moment we are waiting, for this COVID outbreak to stop and like come back to the normal life situation where the entire world is looking for workforce towards India. So I am sure like today for our nation, the need of the hour is new knowledge. And dear friends, new knowledge only comes through research. I would always say, what's research? Research is actually a formalized curiosity is what is research. And it is actually you poke and pry with a purpose. And also to see what all of us have seen. When you think about how when an apple falls, all of us for centuries they have seen what an apple tree, an apple falling from the apple tree, but only one person have thought uniquely about gravity. Such is like what that's what this research is all about. What everybody is concept of seeing completely it for a researcher his concept is different, the way how he thinks like a researcher thinks like nobody else thinks. So this is exactly what is research, I would say. And scientific research, when you say it is a way of life, and science is actually a perspective. Science is also a process that takes us from confusion to understanding in a manner that is very precise, predictive, and reliable. I would tell you, for example, in my own life, I would say yes, there are so many traditional practices at the moment in India where a lot of herbs are used. But unless everything is authenticated with research, with R&D, nothing I never trust. Like I am sure Ayurveda, Siddha, various medicines are there. Unless otherwise it is authenticated and given in a reliable way, if the information is transferred, that's when it becomes a real knowledge. And authenticated knowledge is what now the scientific world needs. So I am sure in India, as you know, there are so many today the pharmaceutical industries are really looking for so many herbs and plant products. And India, you know, especially Coimbatore, with the biodiversity we have, uh, and also so many indigenous species of plants what we have, uh, mm, research in basic science, especially with plants as well as animals, is very important today. Like you need these basics botanists and zoologists for the pharmaceutical company to identify what products can be obtained from such plants, and it can be eco-friendly at the same time, and such things, so on and so forth. So today Parasuraman sir is going to give a lot about the imaging in drug discovery. I'm sure that's going to be quite a very thought-provoking topic he has selected, and I'm really thankful for him during for accepting and helping to enlighten our young minds. Thank you for giving me this opportunity, and thank you, Dr. Parasuraman Padmanabhan sir.
Thank you.
Thank you.
Thank you, madam, for your informative inaugural address. Now I invite our principal, Dr. Lakshmi sir, for offering felicitation to this function.
Good afternoon, all of you. It gives me immense pleasure to facilitate the webinar on Research in Basic Science, a tool for nation builder, jointly organized by Research and Development Cell and Dr.M.Aruchami Research Foundation. The topic is relevant today as the entire world is facing many disasters. As young scientists and research scholars, it is your prime motive to explore the solutions for the natural calamities. At this juncture, one cannot forego the fact that basic science and its applications take prominent part in offering rectification for the entire world, to our nation in particular. I am very happy that Research and Development Cell has arranged this webinar to coincide with the current scenario. I wish the convener to this webinar, Dr. S. Balasamy sir, the R&D research coordinators, Dr. S. Binukumari and Dr. K. Tenmuli, Department of Botany and Zoology, for conducting this webinar, and I wish a grand success. Thank you.
Now I invite Dr. Binukumari Madam to introduce the guest, Dr. Parasuraman Batnaban, Deputy Director, Translational Neuroscience, Lee Kong Chain School of Medicine, Singapore. Now Binukumari Madam will introduce the guest.
Now I take this opportunity to introduce colleague resource person, Dr. Parasuraman Batnaban sir. Parasuraman sir completed his graduation both from Department of Zoology and Anambra University. He has been serving as a Deputy Director, Lee Kong Chain School of Medicine, Nanyang Technological University, Singapore. He has visited more than 30 countries. He has held in different positions such as visiting professor in Alagappa University, Sri Ramachandra Medical University, Chennai, Vellore Institute of Technology, Bharathidasan University. He has editorial role and reviewer in different international peer-reviewed journals. His current research interest in cognitive neuroimaging, multimodal molecular imaging, image processing, regenerative medicine, drug discovery, nanomedicine, viral mediated gene delivery. He has a member of different professional bodies, and he has nearly 15 years of management and networking experience in different institutions and research experience in molecular imaging, molecular biology, cell culture, and microbial genetics. He has received projects from different funding agents. He has published more than 110 papers in peer-reviewed research journals. He has received Young Scientist Travel Award, Young Investigator Award, and Long Service Award at LKC Medicine, NTU. He has received US Patent Award in coated water-soluble quantum dots for stem cells labeling and patent pending in Singapore 2020 in sensing of beta amyloid fibrillation inventors. He has participated 55 international and 35 national conferences. He has conducted 11 workshops and 44 invited lectures. With this short introduction, I once again welcome everyone to this webinar. Now, I hand over the session to our resource person. Thank you, one and all.
Thank you very much for the nice introduction. First of all, I would like to thank the Research and Development Cell, Dr.M.Aruchami Research Foundation, which is timely came out with the idea, Research in Basic Science, a tool for nation building, because India is behind 15 years as far as the experts are concerned. So we need to have more towards the research activities, especially the basic research. First, I would like to thank the patron, Dr.M.Aruchami, Dr. C.A. Vasuki. She has given very nice introduction. I had been to your campus, and which is a wonderful campus, and everybody is working on the basic research and also very interesting research topics. I had a very good interaction with the principal last time, and I also gave a talk during my visit. And also I would like to thank the convener, Dr. M. Lakshmanasamy, Organizing Secretary, Dr. S. Paulsamy, Dean, R&D, Research and Development, coordinators, and I know Dr. S. Binukumari very well, and coordinators Dr. Binukumari and Dr. K. Tenmuri for organizing this nice webinar. This is a timely because everybody is locked in lockdown period, and they feel at least something to have as a, I'm going to talk about the cutting edge. So before I go to start my talk, I would like to request everyone, please turn off, your speaker. So when the question and answer session, you may turn on. And another one thing, so during my slide session, so somebody may have a difficulty to see the slides. For that reason, you have to go in my name and pin the slide, pin the slide, then you could be able to see. So I'm going to share the slides now. Sorry. The sound is okay?
Sound is
It's audible, sir.
audible, sir.
Audible. Can you see my slides? Not yet, huh?
Slides, sir. Slides வருதாங்க சார். Slides வருதாங்க சார்.
தெரியுதுங்களா?
அவரோட ஸ்லைடுங்க சார். சார்.
இல்ல சார். தெரியல.
அண்ணா தெரியல. ஓகே. Now
presentation வரல சார். சொல்லுங்க சார். presentation வரல.
Slides.
இல்ல அவர் வந்து இது பண்ணனுங்க.
I'm doing it. One minute, one second.
Okay.
மீட்டிங் கட் பண்ணிட்டு வந்தா அவுட் சைட் வீடியோ இல்லடா அது பெஸ்ட் நம்மளே லோக்கேட் பண்ணிக்கிறது என்னோட ஜேபிஎல்
Can you see?
Sir, வருதாங்க சார். வருதாங்க.
It is not
visible.
It is not visible.
You wait for a minute.
Oh, okay, fine, sir.
Can you see me? See the slides?
Sir, slide is not visible, sir.
So, what's the problem? No.
இன்னொரு பேச சொல்லுங்க.
So present now, video animation window.
அவர் மியூட் பண்ணிருக்காரு. இல்லைங்க சார், மியூட் ஆகல.
You are not trying anybody, right? So
இதுல கேக்குமா? அதுல ஹெட்போன்ல கேக்குது.
Now
அதுதான் கேக்குறீங்க சார். சார் அவர் வந்து சார் இந்த மெனு கீழே இதை கிளிக் பண்ண சொல்லுங்க சார் அவரை. நீங்க சொல்லுங்க. இல்ல அவர் அது அந்த அப்ளிகேஷன் விண்டோன்னு போட்டுக்க மாட்டாரு.
இல்ல present now I have to share the screen, right? Present now, right? I have to.
அவர் பேசுறது இன்னும் ஆடிபிள் இல்லையா?
present now போயிட்டு அதுல present வர விண்டோ
அது கேக்குது. பேசிட்டு இல்ல நம்ம கேட்கணும். இல்ல அவங்க பேசுறது கேட்கணும். இதுல கேட்கல.
ஹலோ சார். It's not audible, sir.
Chat boxல போட்டுறலாம்ல.
அதுல ஸ்பீக்கர் வருதா?
இல்ல வருதாங்க சார்.
Hello.
ஆமா, அவரை வந்து அந்த கீழே ப்ரெசென்ட் கொடுத்து பவர் பாயிண்ட் ஓப்பன் பண்ண சொல்லிட்டு அப்ளிகேஷன் விண்டோ கிளிக் பண்ண சொல்லுங்க. எல்லாருக்கும் தெரிஞ்சிடும்.
Chat boxல போட்டு விடுங்க.
Chat boxல பார்க்க மாட்டாங்க அவங்க.
இல்ல actually hello sir.
முதல் ஸ்லைடு போடுறேன்னு சொன்னாரு.
Sound வரலையா? இல்ல நீ
So,
சவுண்ட் எல்லாத்தையும்
sorry. I'm going to present now. I'm pressing. Then I have to share your entire screen, right? Hello? Hello? Present now. Now you see? Hello? 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 Hello?
No, sir, it's not visible.
Not visible, huh? Okay.
Yes.
I'm sharing the slides, but why it is not taking? Because I have done many times, but now you should see.
Sir, Velmurugan here.
Yeah.
Sir, Velmurugan here.
Yes.
Sir, நீங்க அந்த பிரசன்ட் நவுல போயிட்டு பிரசன்ட் அந்த விண்டோன்னு பிரசன்ட் விண்டோன்னு மட்டும் கொடுங்க சார். ஹோல் ஸ்கிரீன் கொடுக்காதீங்க, குரோம் கொடுக்காதீங்க.
Window கொடுக்கவா? That's what I
Hello.
Yes.
Sir, 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 உங்களோட your presentation, right?
Yes.
Hello, sir. On your screen, you will be able to see the three-dot structure. Next to that, you will have something called present now.
Yes. That's what I did. Okay.
Yeah, please click. Yes, please click that. And before you click that, you open your PowerPoint in your desktop.
Yeah, I already opened. Yes.
Fine, sir. Now you click present now.
Yes.
You will be able to see three windows. I mean, three options.
Yes,
Chrome
exactly.
and a window. You just click a window and click share.
Okay.
Just click a window and click share. I think we should be able to see your PPT now.
But to share, the button is not working anyhow. I'm clicking the full. Now you see your.
No.
Sir, நீங்க பத்ராவன் சார்.
Yes.
நீங்க அத கிளிக் பண்ண உடனே ஒரு அந்த எந்த பிரசன்டேஷன் நீங்க ஓபன் பண்ணி வச்சிருக்கீங்க அதை செலக்ட் பண்ணிட்டா தான் ஷேர் ஆக்டிவேட் ஆகும்.
அத செலக்ட் பண்ணி ஆக்டிவேட் பண்ணுங்க.
உங்களுக்கு
click that presentation.
அத செலக்ட் பண்ணாதான் உங்களுக்கு ஷேர் பட்டன் ஆக்டிவேட் ஆகும்.
ஆமா. இல்ல சார், அந்த விண்டோல அந்த உங்க பிபிடி தெரியும் பாருங்க.
ஓகே.
அந்த நீங்க ஷேர் பிரசன்ட் நவுன்னு கொடுத்தீங்கல்ல சார்.
ஆமா.
அதுல நீங்க ஏ விண்டோ அப்ளிகேஷன் விண்டோங்கறத கிளிக் பண்றீங்க இல்லையா?
பண்ணிட்டேன். ஓகே.
அத கிளிக் பண்ணா ஒரு பாக்ஸ் ஓபன் ஆகும். அந்த பாக்ஸ்ல உங்க டெஸ்க்டாப்போட எல்லாமே காமிக்கும்.
தெரியுதா பாருங்க.
அதுல உங்க பிபிடி தெரியும் பாருங்க. யா ஃபைன், இட்ஸ் கமிங் நவ. Yes sir, fine. Yes.
Sorry.
Yes sir, அவ்வளவுதான். You can go ahead sir.
We use Zoom, not me. Okay, anyway. Sorry for
sir, Zoom வந்து போய் use give PPT. Full PPT போட்ருங்க sir. You can it's fine sir, it's fine. Perfect.
Okay. Thank you.
Okay sir. Thank you.
Okay. Today I am going to talk about the role of multimodal imaging in drug discovery. The imaging is one of the cutting edge. Like molecular imaging, the omics data, and also the translational platform. The translational, that is bench to bedside. how the basic research can be translated into bedside. So these are all the new buzzwords, but it is there already, but people are started, and now they are introducing even any kind of the grand application you want to apply now globally, you have to put this kind of translational research, which is more regarded like anything. So here I want to put the molecular imaging, the first slide. The molecular imaging is basically it to understand the molecular cellular processes in vivo. For example, if any cellular events you want to look at, but you cannot document, for example, the biochemical data, sometimes you able to give only numbers, which is not evident to document the cellular processes in vivo. So for that reason, the molecular biology and imaging, the amalgamated form, they came out with the molecular imaging. So you can able to document and visualize by the cellular activities or cellular processes in vivo, and finally you could see where exactly you are getting the signal or the activities endogenously happening inside the cell. So here this cartoon shows the molecular, especially the cellular molecular level. For example, you want to target an organ which is secretory in function. The secretory gland normally have more Golgi complex in it. So your target should be more secretory in nature. That means your biomarker or your biomarking approach should be started in that manner. So the in vitro, globally, the basic laboratory used to have the in vitro culture facility. By the way, we can test in vitro level. That means outside the same cells may be grown outside the environment, outside the body or the organism. And finally, you could be able to document or account the cellular processes. So here we have to be very careful. So when you are doing this kind of the assays, so whatever you are getting, like a several fold increase in vitro setup, then only you can go to the in vivo. For example, if you get an 8-fold increase in the in vitro assay, that means that something will happen in vivo. The reason is manifold. The one simple reason I want to tell you is that the living system has the excretory system. The excretory system and circulatory systems are the common thing where the molecule or the biomarker, it circulates inside the body, and normally there are so many factors involved, and finally whatever you are seeing in a stagnant in vitro culture, which will not happen inside the body. So 8-fold increase means you could see only in vivo 2-fold increase. That means, so the system is very complicated. The living system will have many things, including circulatory system, but the stagnant system like in vitro culture don't have such kind of thing. So that is a key. Then, so when you want to do that in vitro is you get a 8-fold increase, for example, then you can go to the in vivo. In that you can start from rodent and end up with the non-human primate. Non-human primates are nothing but the monkeys, which are close associate. After the human genome 2000 project, we could able to understand the human genome versus the other organism, the experimental organism what we use from even vertebrates, invertebrates, everything. So the non-human primate, especially the monkey, which resembles the human activity 95%. So that's why the final FDA, Food and Drug Administration, demands the monkey data. So that means you have to have the non-human primate data as a final data. Then it will enter into the phases, several phases. That means 4 drug discovery phases: phase 1, phase 2, phase 3, and phase 4. And I will come in a minute, about that. So here, so what is the imaging? The imaging is an integrated platform. Why? Because there are the discipline person is essential, or the coordination is important, or the synchronizing work, should be followed. For example, if the doctor has, doctor only, he can look into the improved health. What it means improved health. For example, in the hospital, US, the doctor also do research. So by the way of research, he tried to improve the health of the human being. How? Because the practice, there are so many medicine in practice, but the doctor will experiment some more medicines or the same medicine in a different combination with the patients by doing the research as well as treating the patients. So that means the improved health will be seen. That is why the country like USA normally used to have, or that normally the doctors do research as well as the practice. So which is very essential. So the why it is integrated platform, without chemist, you cannot synthesize a molecule. That means that molecule is essential for biomarking. That's what. Now the biomarker is the buzzword, which is, biomarking will be done, or the biomarking exercise will lead to the drugs. So that's what we are doing. So this is what the imaging is the integrated platform, which is very important. And the history of imaging. So here I want to emphasize, because it is not an easy job. For example, the Roentgen discovers X-ray in 1895. And in 2000, my good friend, Professor David Townsend, he invented PET-CT. He is one of my mentors during my the course of my development. So the Professor David Townsend put the PET, positron emission tomography, with the computer tomography, which took 100 years from the Roentgen. So what it implies, because it is not an easy job if you can able to document something, because a computer tomography, of course, the information technology is a boon nowadays. It helped us in a way to evolve how, the digital world. That is what we are comfortable. Even this lockdown period, we are communicating each other and we are surviving. And here there are preclinical. Before I go into the clinical side, so I want to emphasize the preclinical. So the people may think, the researchers, especially, we know that the instrument, for example, positron emission tomography, which is in the hospital setup, the similar positron emission tomography is possible in the small animal as well. That is called preclinical setup. So these are all the preclinical imaging equipments. It is similar to the imaging equipments which are available in the hospital. For example, the micro-PET, which is the miniaturized version of the PET, positron emission tomography. And the autoradiography, that autoradiography, normally it helps only on the postmortem sample. So you can imagine the postmortem is only possible in the small animal. Not at all. You can do postmortem samples, the probing in the human sample as well. So like that MR, and you have ultrasound, microspect. SPECT is a single photon emission computer tomography. All are available. To maximize this thing, the hospital setup, the imaging modalities for the human, we have X-ray. X-ray is nothing but our good old like a photography technique, which is 2D, that is a snapshot. You can able to see. But still, this is the golden standard nowadays, because if anybody wants to have a job, first place, the medical examination will be done. For the tuberculosis, still we need to have the X-ray, that is the 2D, but it is the X-ray. The improved version is the CT, because computer tomography and several these snapshots merged together because of our technology, digital technology. Then it is you can able to see 3D. That is a 3D render volume, everything possible. And also MRI, magnetic resonance imaging. FMRI is the functional magnetic resonance imaging. By the way, you could able to see the brain function. And nuclear imaging is two equipments you can use. One is the PET, positron emission tomography, and another one is single photon emission computer tomography. And ultrasound, normally, it is everywhere. In any hospital you go, you could see the ultrasound, because it is a mandatory equipment to even for the OG department, obstetric and gynecology department. So these are all the imaging modalities available in the nutshell. So what it is implied. So what is the use of these things? For example, here I want to correlate these equipments with the drug discovery, which is very important. So why? Because now Food and Drug Administration, they don't want to believe the data given in numbers. They want to believe by, what is seeing is believing. So they want to see the data sets which is in the image format. For example, if we talk about the sensitivity of the methods, I will come in a second. So how the imaging modalities are more sensitive, and how it is to be accounted in the setup. So here, this is the type of imaging. So what I have shown before, these are all the modality which are available in the hospital. So now I want to say what it is good for, these modalities. What you can get out of it, these imaging modalities. Here, the CT, the computerized tomography, and the computer, this will give only the anatomy. That's why if any bone scan you want to do, the CT is the best option. And physiology you cannot see, but you can see little amount, but in the soft tissue contrast. If you inject some soft tissue contrast, then you could see some physiological parameters. That's why if you do the angiography, when you go for a heart disease, then they will inject the contrast agent and you could see the physiology. This is the one classical example I would like to add here. And ultrasound, this is also good for anatomy to see. And MRI, of course, you can see anatomy, physiology, metabolism, but not the molecular level details. But you could see only a small amount. So, but at the nuclear imaging, which is very good for physiology, metabolism, and molecular level details, but not good for the anatomical details. That's why the hospital setup, thanks to David Townsend, my mentor, he just put the PET and CT together. That way, it is good to understand entire anatomy, physiology, metabolism, and molecular level. So that is it good for. And optical imaging, of course, it is good for molecular level details, but we cannot translate the results to the human setup, because whatever the fluorophore we use, which are considered to be a toxic, and it should not be injected into the human, and we cannot experiment with the human body. So what they are doing now, they want to improve the optical fluorophores, which, by the way, they want to come out with the toxic free chemicals, and they want to implement. So these are all some glimpse. And the multimodality imaging, then I want to give a small detail about what this is good for. MR imaging, the machine used powerful magnetic field. We talk about the Tesla. So 1 Tesla is equal to, the Earth also we have a magnetic field. That's what we say, that north side, the magnetic attraction of the Earth is more. But if you consider that, it is 3,000 times more than the Earth magnetic field, which we consider as the 1 Tesla. So then high cost setup, high maintenance. Optical imaging is a very cheap, but because we cannot translate. But micro PET imaging, which is very good, but it is also high cost setup, and it is the clinical applicability is very high. And handling radioactivity, because some of the people, they don't like to use the radioactivity, but the precision is very good actually. That is why even in olden days, if you do some radio immunoassay, which will be accepted in a high way, because the results are considered as better. It's a very highly sensitive. That's what, even if you want to publish a paper, those days you have to do the radio immunoassay. The multiplexing, multi-pinhole SPECT, single photon emission tomography, which has multiple pinholes. Through the pinhole, the resolution will be seen, and the results will be obtained. But what is the difference in the nuclear medicine PET and SPECT is, the PET is the high energy, because if you look at the molecule, the radio tracer you are using, which is 500 and above, 511 and above kilo electron voltage, whereas in the SPECT, the energy level is low, which is below 300 kilo electron voltage. So which is the difference. So you cannot image with the same camera. So one is a very high-end camera and another one is a low-end camera. So that is why the multiple pinhole the apparatus, which is called SPECT, single photon emission computer tomography. So here the need for translational research. I already told you I'm going to use some buzzwords. So what is the need for translational research in general? For example, if you want to have a drug discovery, So definitely you need to have a translational platform. So what it means, whatever you are doing, the basic molecular synthesis, molecule synthesis in the chemistry lab or in the biological lab, that biological molecule should be, validated by means of different stepwise procedure. For one good example is now after 2000, the genomic project. So we found out 200,000 the disease modifying genes are available. Our IT professional, especially the bioinformatics people, they analyzed the entire data set and they came out with the 200 annotating genes. That annotating genes are very important to modify the disease or to cure the disease in the human being. So the question is asked by many experts and scientists. So whether we do we have the drug for entire 200,000 annotated genes? The answer is no. How much we have? We have only 10%. So that means we have a duty to synthesize or find a drug for remaining 90% of the annotated genes or disease modifying genes. So in which way you can do it? For example, the omics platforms, the genomic, proteomic, metabolomic, and even transcriptomic platforms will be helpful to identify such a biomarkers. And by the way, we can able to, will be successful in finding the drug. So on the right hand side, I have given the disease modifiers as an example, citing that the Human Genome Project. So what is these gene modifiers are good. For example, any experiment you do, we do the control experiment. So here, the same way, the panels clearly indicate that. So you see that where this is the dopamine transporter in the brain. The dopamine transporter especially targets the dopamine receptors, which are in the striatum region of the brain. So what you are seeing on the left-hand side, there is a red, there is a highlight, there is a emission of the radioactive substance. What it is targeted to the brain in the striatum region is it is called FEP2I, which is a tracer for this particular dopamine transporters. So that means it is targeting only the striatum regions. So that's why you are getting a very clear signal here. And whereas in the right hand side, because we block the entire signal, so that means we need to have a control whether the molecule is the biomarking exercise is complete, whether it is targeting the striatum region, especially the dopamine receptor. That's what you are seeing. We entirely blocked, and you are not able to see the signal there. That means the receptor is completely blocked, so there is no signal there. So the other one on the top, it is, like we have given some medicine orally and we have done the PET studies. So translational research need, when we talk about the drug discovery, so the translational research is very important. So what is the the researchers should design and concentrate on bench to bedside research. So the translational research is nothing but bench to bedside. So that means, for example, in our country, especially in India, so many people are working on the nanomedicine. So many people are talking about the drug delivery. So many people are not even knowing that what is the microenvironment of the tumor, and they try to deliver the drug there. So these are all the things. So how it can be, translated to bench to bedside. So you have to have a strategy. For example, if you want to, one good example I will tell you is that if you want to target the tumor, the microenvironment of the tumor, the pH level is lower than the normal tissue. That means you need to design the nanoparticle, that is whatever the functionalization should be done in such a way only the drug should be released in the pH specific manner. That means in a lower pH. That means you can deliver very efficiently. The rest of the area, even though the particle circulates, it never ever releases the drug. So that is the one of the thing. So that may be translated. And one another thing is that every time we just test in the in vitro culture, then we forget about the particle and we do another research. So we have to take it to further. to the rodents and non-human primate and further to the human being clinical trials. And genuine reasons, the technology and applications will promote the collaborative research with other groups, can contribute to translational research, information technology, material science, chemistry, biology, engineering, etc. And finally, I would add the economic reasons, because no one is working in the world. The final destination, especially everybody want to see the outcome as the money, because the economic reason. So we have to do the translational research. Once a single, even though you develop like millions of molecules, and finally you end up with one molecule which will, like control some drug, very specific disease. That means you will be your entire. That is the gold mine. So that's what we have to aim. And NIH spending. So this is the particular and also so important considering the basic research, especially here. So I want to put it the NIH spending how, because not I'm putting the world data in 2008, how the NIH spending on the clinical research. You see the non-clinical research is around 67%. So what it means is that, so NIH is more focusing on the basic research. The basic research only will, end up with the clinical trials. So that's what, if you see the clinical trials, only 12%, whereas the basic research, the non-clinical research, which is 67%, which is around 19.8 billion every year. So, which is very evident that the basic research only very important in any developed or developing nation. So it is also our bound duty as researchers. So we have to think in a way how this the basic research will be translated into the bedside. That is means in the clinical setup. And also the translational medicine platform. There are many platforms. You see that molecular imaging is occupying the third in position, but now it is moving to the second, because bioinformatics era has gone, because we already identified. Now the NIH is asking more on the PET data, because any drug discovery has been done. So immediately the NIH is asking, where is your PET data evidencing the claim? So that is what happening now. So it moved towards the first level. So that is what it is. And also the preclinical and the clinical imaging and the preclinical imaging. So in olden days, for example, you want to have the experiment, you need to have a different batches of animal. For example, you start with the rodent. So you need to have 10 batches of rodent animal. So every time point you have to kill the animal, and you have to do some autoradiography, or you have to remove the organ, you have to do the histology, histochemistry, so on and so forth. But now, because of this molecular imaging, so you don't need to do anything. So the same one group of animal, you can do imaging longitudinally with a different modality, and finally you can account the data sets and how it is, whether it is controlling the disease or it is, progressing, the disease is progressing. For example, if any animal model, so you have to develop in the case of tumor model, you have to do the xenograft first to be made. After 15 days, it started growing. Then finally, the growth will be monitored. What is the size and other things. So by the way, this is the way to progress. So this is what it is. So, and translational imaging, that is what I put earlier, in vitro, in vivo, and clinics. And potential areas, cancer, gene therapy, drug screening, xenotransplant, that is the organ transplantation, neurodegenerative disease, metabolic disorder, toxicology study, all the disease can be tested. But here I want to emphasize that this is a faster screening, and you can follow them longitudinally. By the way, you can able to document what result you want. Exactly. You start from 6 months and you end up with the research, because rodent only survive 24 months. So you can track them up to 24 months and you can document all the results of time, and you can able to conclude whether this compound is feasible for going for the clinical trials. So the next one is the drug development. I want to put a small the thing. So how the drug development has been done, and because the pharma companies normally synthesize like millions of compounds, and finally the CRO, especially the contract research organizations now everywhere, like so many drug companies and contract research organizations, the multinational companies, they are willing to do your compound, screen your compound. That is called high throughput screening. By the way of high throughput screening, then they will find out the lead molecules. What are the lead molecules available that the lead compound can be tested further. But for that, even for this, it may take 3 to 3.5 years. Then you have to go for a clinical trial. So that this 3.5 years, it started from the synthesis of the molecule, then the in vitro study, then preclinical started with the rodent and monkey, which will take 3.5 years. Then after that, you have to do the phase 1 trial, phase 2 trial, phase 3 trial, and finally the phase 4 trial, which is a post-marketing testing. So that's why some of the drug, even into the marketing, then they will withdraw. That means post-marketing survey, which is a very essential, whether it have some side effect and other things. So by the way, the drug discovery will be done. In earlier days, it took nearly several years, 15 to 20 years. Now, because of the multimodal imaging, it cut down to the 7 to 10 years. So which means the longitudinal studies are possible, and because of that, we could be able to achieve the level of, we reduce the time. We don't want to wait. Even the COVID-19, what we are doing, we want the globally, they announced there is no institutional review board approval is needed for doing any COVID-related research. What it means is they want very quick the medicine. So that's what the people are doing. So it is very good if we have this translational platform to do. And here, the one classical example, how the translational platform is and what are the things to be needed. So here, what you are seeing exactly the entire complete translational molecular imaging platform. So what you are seeing on the left-hand side, the brain. So that means, so I am translational neuroscience. So our team mainly work on the translational, especially neuro-related problems. So how the neurodegenerative disease, the early biomarking approach, and finally how we can get rid of the neurodegenerative diseases. So one good example, or model we have to use is that post-mortem samples. So for that, we have a brain bank in our school, the medical school. So what we do is normally the people sign up for the brain donation. So those who are diseased, so normally we fill the form. Immediately once the person is died, the next kith and kins, they will inform us, and before 6 hours we will remove the brain and we slice them and keep it in the bank. Because if they are neurodegenerative disease affected, some disease like Parkinson or Alzheimer diseased person, so that is really a good, the sample, the material we preserve. And whenever this kind of research we do, we just use the brain slices as our sample, and we do autoradiography and other things. And the other way that we use that micropet is a one good example I put here, one imaging modality. And first, the brain bank, we obtain the brain. And what are the probe we synthesis? For example, some radioligand. We probe them and do the autoradiography. So autoradiography is a very old technique, but still it is the golden standard. Then we have some hits or some good results. Then we process them and we test the same molecule in rodent and non-human primate and human subject. So if you are starting from the in vitro and you are finishing with the human, which is called forward translational research. So you are starting from the scratch and taking further and ending with the human. For example, if you are starting from the human, that means the post-mortem samples, and immediately you are injecting into the human being. That's what the COVID situation, most of the research, the teams, they try to understand and they try to do it. So the reverse translational research, that means they will go from the backward to forward. So that way, you get the good results. Okay, we test in the other animal, same species, it works, or different species, the same molecule worked for the different species as well. So that is what it is. So here you see that the left-hand side, what you are saying is the autoradiography, when you probe them and finally you take the picture. And the middle, what you are seeing is the small animal imaging platform, which has the MR, SPECT, and CT. These two machines have interchangeable beds. The same animal can be changed to the different machines, and finally you get all the details about the SPECT, PET, and MR images. And in the right hand side, which is the higher sensitivity human scanner, which also can be used to test the same thing. So what you are seeing here is the rodent. You see the striatum region is lighted up in the monkey, and also in the human brain you could see the signals. That means it is all working from the the forward way from the rodents to the human being. So here, the possible contribution of various translational medicine research platforms. So I already told it is a multidisciplinary approach. So because we thought that in NTU, the Nanyang Technological University, how the different school we can bring, because being a medical school, so our facility is where, we can do anything, because from the rodent to human being, any kind of, the animals we can test. For example, we have rodents, we have even marmosets, which is called New World monkeys. So we are breeding them in our facility. So that what is the difference of the monkeys and marmosets? Both are the primates, but this primate lives only with the siblings. It mimics the human nature. So that's why now the scientists are preferring marmosets instead of the monkeys. The macaque. This is the marmosets are the New World monkeys. So here you see you can do all the things like omics and disease models. We can create our own models and behavioral test, imaging, pharmacology test, all being a medical school we can conduct. And you can see in the different the platform, especially different schools, they come closer because we are collaborating almost all departments in the entire NTU premises, because everybody want to cross-pollinate, and they want to conduct at least they want to include biology part. That way they can publish and flourish also. And the molecular biology receptor imaging. So now the question is, so if you are having a drug discovery platform, so what it is good for? So how this molecular imaging is helpful? Because the one good example is that, so I want to give whether the question is whether the receptor can be imaged by molecular imaging. So this is the one example. So this is the estrogen receptor alpha. The estrogen receptor, everybody knows, it forms the nuclear family. It belongs to the nuclear family. So it forms the homodimer. So what it means, if any ligand it finds, for example, the estrogen or the drug like, the any tamoxifen, relaxifin, hydroxytamoxifen, these are the drugs to be treated for the breast cancer. So if you using this kind of the ligands, so whether how these the receptor behaves, so that's what we want to understand. For that, the molecular biology, especially, you need to have the gene constructs. So what is the gene construct? For example, the cell. So cell has different organelle. So every organelle inside the cell is defined for their function. So for example, endoplasmic reticulum is a smooth and rough endoplasmic reticulum. The rough endoplasmic reticulum has a ribosome where the protein machinery happens. So like that, there are several, every organelle is defined for a specific the function and also specific activity. So here, so especially every cell, it has its own endogenous activity. For example, the cell maintenance. The endogenous activity is routine, and it performs their endogenous activities without a, it is on and off, because there are switches inside the cell. It will work automatically in a normal human being. For example, if you want to have especially something, the report, the cellular event, how you can document. For that, you have to use the reporter gene. So reporter genes are nothing but reporter gene, reports cellular activity in vivo. Because there is endogenous activity, forget about endogenous activity, because now you are introducing a gene which has a reporter. For example, firefly luciferase. That means which is give light. So that kind of reporter gene which reports the cellular event in vivo. So when you are injecting the D-luciferin, the substrate, immediately the light will be produced from the cell. So that's what the reporter gene. Reporter gene reports cellular activities in vivo. So now, here we just formulated and we made a construct, and ERE, that is estrogen receptor responsive element, alpha, and positive and negative, and we made a construct, and finally we just added in the cell line and we tested. For example, here for the estradiol is the they activate the homodimer, especially the estrogen receptor. In the presence of estradiol, it forms a homodimer and it binds with the estrogen responsive element, and it translate and transcribe and translate into protein. That's what is happening. So now this cartoon, just there is no binding of estrogen in the receptor. That means there is no homodimer formation. So now what the estrogen binds with the estrogen receptor, then it forms the homodimer, then it binds with the estrogen responsive element, and it transcribe and translate into protein, which is the firefly luciferase, which we endogenous, that means reported, we send as a reporter, we transfected the cell, made this table transfection, and we documented. So now this is what the renilla luciferase. Renilla luciferase is the luciferase which has the kinetics of 1 minute. That means the moment you inject the substrate, and immediately it will give the light. So that is what we are seeing here. If the estradiol is not binding with the receptor, that means there is no formation of the homodimer. If it binds with the receptor, it forms the homodimer and binds with the estrogen receptor, the estrogen responsive element, and finally it gives the signal. So on the basis, we use this is a split reporter concept. Whenever these two split reporter come closer, then it gives the signal. That is the concept. And finally, these are all the materials and methods. So what I want to emphasize here is the homodimerization formation. So for that, you cannot decide on with the simple single reporter gene. So you have to make several permutation and combination, and you have to test them, which is giving more homodimer formation. So for that reason, we selected, this is the one which gave very good result. That construct is used throughout the study. And finally, we treated with several, ligands in the 293T cells. That means this is a embryonic kidney cells line. So here, this is the control. Then tamoxifen. Tamoxifen is the cancer drug, especially breast cancer treatment drug. Relaxifin also the drug. Hydroxytamoxifen also a drug. And genistetin diethylstilbestrol, cisplatin, or and estradiol. And this genistetin diethylstilbestrol, these both are the semi-agonist or antagonist, and cisplatin is the drug. This is a cancer drug. So we want to test the cancer drug as well. So that is what we are, did. And finally, you could see which nanomolar concentration it works. So we have to decide the dose, because whenever we treat the patient, or the dose is very important. That's why we finally we could able to see the thing, and we concluded that the tamoxifen and the relaxifin and the estradiol. This is estradiol is activate, that means the promote, and the other one, the tamoxifen and relaxifin, it suppress the disease. That is what the activation and repression. So that's what we find. So estradiol, so any the protein conformation, so it is actually the conformation, it one, the estradiol it promotes, but the other one, the tamoxifen and relaxifin, it suppress the disease. That means it, one is activation and another one is the repression. So that is what we observed. And finally, we tested this. When you are putting the reporter gene, you have to confirm whether the reporter gene is intact inside the cell. For that reason, we just did the normal Western blot assay. That is whatever they put, and we could see as a different fragment inside the cell. So that is what to document the Western blot has been done. And finally, we did the imaging. So this is the conclusion, will be utility of the preclinical evaluation of any drug. So for example, you synthesize a molecule, you want to test this against the cancer, especially breast cancer, especially the estrogen receptor alpha, you can use this system and you can test rapidly and you can find it. This is another one. For example, the 5-fluorouracil is used here, and the cancer model has been developed, the xenograft model. And even on the day 3, you could see the suppression in the tumor volume. So which is the indication of how this animal model will help to evaluate the drug. And the nuclear imaging, especially I told the nuclear imaging is the 2 imaging modalities. Here one is the SPECT single photon emission tomography, another one is PET positron emission tomography. The single photon emission tomography here, you can even see the motility in the problem. So there are some GI tract motility problems or GI tract problem. So for that, so the mice has been selected, and we just gouged with the charcoal along with the ligands, especially radioligand. So you could see this is the snapshot of different phase, how the radioligand traveling in the GI tract. And there is, if any, there is an injury or any problem in the GI tract, you could able to, see the higher activity in the particular region. That is what you are saying. This is the SPECT image. This is the maximum intensity projected. That is the first one. And coronal view, sagittal view, and transverse view. And here you can see that clearly where exactly the GI tract, the concentration is more in the particular lesion area of the animal. So again, so these are all the machines which I have shown earlier. This is a PET CT and PET MR. This things are interchangeable. The beds are interchangeable. So you want to do PET and MR, the same one machine you perform, and you can change the PET, the same bed, without shaking or moving the animal, just to transfer to the other imaging the platform, especially the machine. Then you perform the study. So that way you can get you can fuse the 2 images, the PET image and also MR image, or PET image and CT already there, and PET image versus MR, you can do that. Or sometime you want to do the CT from that the machine to this machine, you can transfer the animal and you can perform. And this is the same. And here, this is the maximum intensity projected view of the mouse. Here it is a 99M. The pertechnetate is used, which is the SPECT agent. If you inject, what is good for pertechnetate? Pertechnetate, normally it is used in the clinical setup, especially for the thyroid, the testing. So why? Because this is, the sodium iodide symporter. The sodium iodide symporter is more in the thyroid region, especially in the thyroid. So the pertechnetate, especially the technetium 99M, that is another name for that. So will be injected. And finally, you could see only in the thyroid portion as well as in the stomach. The stomach also, we have the sodium iodide symporter. So this is the maximum intensity projected image with the CT. This is a CT with the SPECT. So these two images were merged, and you could see where the exact position of the thyroid and the stomach. And NIS, that I already told, sodium iodide symporter is a membrane glycoprotein mediating active iodide uptake in the thyroid gland, and it is the molecular basis for radioiodide 131 imaging and therapeutic ablation of thyroid carcinomas. So what it very good, actually, when the thyroid patient, especially thyroid, there is a tumor in the thyroid, what normally they do, they will surgically they will remove, but they cannot remove because that area is very sensitive. You cannot do that, the complete removal of thyroid. You can do certain percentage only. So then after that, in order to ablate, they give iodine 131 in a tablet form. It is so that every hospital is now in practice. Then it will ablate the cancer. So what is it good? Actually, here, see, what is the thyroid, especially the sodium iodide is important does. So 3 sodium, first it will come from the thyroid cell, and 1 iodine will be guarded by 2 sodium molecules. So that for that, they want to get 1 sodium inside, the 3 sodium first they will come out, and they take in 1 sodium with 2, take 1 iodine with the 2 sodium molecules. So that's what the cartoon you are seeing here. Now you clearly see 2 sodium it taking 1 iodine inside. That is why this is called sodium iodide symporter. So it symport the iodine molecule inside the cell. So this is the cartoon well explained. So how the process happened. But the molecular imaging by the PET imaging only it targets the thyroid, especially if you inject the BF4 in the mouse, and it clearly targets. This is the image which is superimposed to the CT image. The PET image imposed to CT image, then you clearly see the signal coming from the thyroid. So which is the beauty of molecular imaging. That is the reason why the FDA, the Food and Drug Administration, is more asking nowadays the PET data, which is very authenticated. You can even tell how much is the uptake in that particular region. The machine will calculate everything. You inject this much dose, you give, then it will calculate all decay, because radioactive substance will have half-life. All the half-life will be calculated, and finally you can say that what is the uptake, the amount in the particular lesion area, or in the tumor area, or in the particular organ. So again, the upconversion nanoparticle, you can use for the therapy, photodynamic therapy and other therapies. So these are all the one of the study you can be able to conduct, and you make a tumor model and deliver them, and finally you can do MR or PET, that kind of modalities. And here the peptide coated quantum dots that we targeted for damaging the DNA inside the cell. By the way, you can disturb the DNA, that means you can kill the cells in principle. That is what the patent we filed. So we could clearly demonstrate that we target the nucleus, and because targeting nucleus is not easy, because when you are any the nanoparticle, if you are incubate in the cell, you incubate in the in vitro culture, then what it does is it cannot, it normally it won't allow, because of the cargo mechanism, it won't reach to the nucleus. So by the cargo mechanism is so complicated, and it won't allow any foreign particle get into the nucleus, because that is the vital portion of any cell. So somehow we made a the coating, and it finally it reaches to the nucleus and they kill the cells. So these are all some of the results. And you may ask some other question also, because considering the interest of the time, I didn't show many data what we are doing now, and what is our center is equipped with. And I think in another opportunity, I may come up with the rest of the the research what we are doing, and what are the drug we synthesize. We recently synthesized 17 small molecules, which is curcumin based, and it will be targeting the brain, especially the tau protein, because tau is the main Alzheimer's disease, because Alzheimer's disease mainly happen because of the amyloid precursor protein, tau, and tangles. But the tau is not very well studied because phosphorylated tau. So we are targeting the phosphorylated tau, and we synthesize 17 compounds. Out of 17 compounds, and 3 compounds are working well, that we are doing and started doing with the brain bank help, and we probed, and 3 compounds are working well, and we are doing further research on it. And I think if some point, if we have chance to come over or chance to have another talk, and we can able to I can able to share the details of the thing. So I would like to thank my mentors, Professor Balash, who is the Nobel Committee member, and Christopher Haldane, who is the Karolinska chemist, who more than 2/3 of the radioligands were produced in the world, and ex-chairman Sir George Rada, who is one of the pioneer in doing the especially the MR inventors, because he is not given Nobel Prize, but he is the one among the person when MR, the Fourier transform, was done. He is the person dynamically, actively involved from the Oxford. That is why you see that the magnet, all the magnets are Oxford made, even still now. And Professor David Townsend, these are all my mentors, all are mentors. Professor David Townsend is a PET-CT inventor, and I had opportunity to work with him, and he is now settled in Canada. And of course, I went to Stanford, where my entire the molecular imaging was taught by Sam Gambhir, who is the pioneer in the molecular imaging. And of course, my colleague, Dr. R. Paul Murugan, I would like to thank in this occasion. Thank you very much for your attention, and I'm happy to take your questions. Hello?
Yeah. Hello, sir. Sir, thank you for your excellent, wonderful, and informative lecture about the multimodal imaging in drug delivery. So we are very fortunate to hear such a wonderful lecture today.
Yes.
The autoradiography, role of autoradiography in the pre-clinical trials and the clinical trials, and the recent technology like PET analysis, everything is very good, sir. And we are much appreciated about your talk and the content there. So now it is open for discussion. The participants can ask questions and unmute your mic. Thank you. Now participants can interact with the speaker.
Hello.
Yes. Yes, sir.
Sir, good afternoon, sir. I'm Karthik from Kongnad Arts and Science College, sir. I'm second MSc Biotechnology student.
Yes.
Sir, my doubt is what kind of advanced nanomaterials used for the basic imaging techniques, sir?
You can use any nanomaterial. For example, if you want to use the carbon nanodots, even carbon nanodots. If you have functionalized that carbon nanodots with the fluorescence or luminescence, I think fluorescence. Normally the carbon nanodots fluoresce, right? So you can use fluorescent, that in vivo, that called there is an in vivo system which can even image some tumor model or some other things. Or otherwise you can use the iron oxide nanoparticle. The iron oxide nanoparticle is you see many papers. You can use MR to study the fate of the iron oxide nanoparticles. So you have to functionalize in such a way. Even multifunctional functionalization also possible. For example, the same particle you can even tag some of the gadolinium. Gadolinium is the positive contrast for MR, and sometimes you can use some fluorophore in order to do some fluorescence imaging. Sometimes even you can use as a PET labeling, positron emission tomography. You want to do, you can even tag them. The only problem is that you cannot use this particle for the brain imaging, because it is a problem to cross the blood-brain barrier. So otherwise, if you target the tumor and other organs except brain, you can target them.
Okay, thank you so much, sir. Sir, and one more doubt is it's only based on the compatibility or the charges, sir?
It is not charges actually. It is a compatibility because it should be biocompatible. That is the first one. Even though we are doing MTT assay for the particles, but it is only certain extent you can tell in the in vitro system. But if you are putting in the same particle into in vivo system, which may behave differently, because I already mentioned in my beginning of the talk that in vivo system has a defined circulatory system, defined excretory system, and other things as well, immune system as well. So because certain extent it is very difficult, but at the same time we can be able to control using some of the pro and con. How we know from the experience, you can learn from your experience. Yes.
Thank you so much, sir.
Yes.
Any other questions from audience side?
Hello.
Sir, one more.
Sir.
Yes.
Sir, this is Katreshwari from Zoology Department.
Yes.
So actually, my clarification doubt is how our body considering that nanomaterial is non-antigenic, and how we can find out the non-antigenic property of that nanomaterials or nanocompounds.
Yeah, that is why we all use the buzzword biocompatible. So first of all, we have to the particle should be biocompatible. So that is the first thing whenever we start doing our synthesizing our particle, we have to keep in mind we should be biocompatible. The second thing, when you are functionalizing, for example, some people want to target some of the using the antibody and other things. There are problems in the immune system which is very difficult to handle when you come to the live animal, especially living system. So we have to be, that is what, we have to plan in such a way. So how it will be, you have to go through the literature. Again, it is not easy, but you have to go through the literature. The first thumb rule is that it should be a biocompatible when you are using the material. Yes. I don't know, I answered your question, but yeah, this is
yes,
the one.
Thank you, sir. Thank you.
Any other questions?
Sir, one more question, sir.
Yes, sir. Yes.
Sir, the nanoemulsions also take the same character as well as the nanomaterials?
It is what kind of emulsion you are using. That is also important. I don't know how the chemical. It is all depends on the chemical nature. What kind of chemistry you are adopting. That it's all depends on that. And also sometimes the cell even it doesn't mean that if you are using, for example, kidney cell line, it behaves. The other cell line will behave differently. For example, neuronal cell may differ differently. So that is what you have to, experiment this as well. Yeah.
Thank you so much.
No.
Shall we finish the program?
Yes. Okay, sir.
Fine, sir. Batmanam, sir, very good talk we received today. Now I invite Dr. K. Thenmuli, Assistant Professor of Botany and Head of the Department of the Botany, Konganadu Arts and Science College, to propose vote of thanks.
A pleasant good evening to one and all present here. I feel very much humbled and privileged to propose vote of thanks to this distinguished and intellectually stimulating gathering. At the outset, I record my deep sense of gratitude to our most esteemed President, Dr.M.Aruchami sir, and Dr. C.A. Vasi Madam, Secretary of our college, for their unstinted support and inspiration. With great pleasure, I deem it privilege to convey my gratitude to Dr. M. Lakshmanaswamy sir, Principal of our college, for his wholehearted and enthusiastic support. On behalf of Research and Development Cell and Dr. M.Aruchami Research Foundation, I submit my sincere thanks and respectful regards to our today's resource person, Dr. Parasuraman Batmanabhan Sir, Deputy Director, Translational Neuroscience, Head of Operations Center for Neuroimaging, Research at NTU, Research Administration and Support Services, Lee Kong Chain School of Medicine, Singapore, for sharing us with his kind words of wisdom on role of multimodal imaging in drug discovery that radiates us with a source of energy within us. Thank you for your enlightened and thought-provoking words, sir. Next, I deem it privilege to thank Dr. S. Palsami, sir, Dean, R&D, for his instinct support and encouragement. With great pleasure, I wish to record my special thanks to Dr. V. Chinnasamy, sir, CEO of our college, Dr. S. Madan Shankar, sir, Dean Academics, for their wholehearted support and enthusiasm. Last but not least, I accord my sincere thanks and gratitude to all the participants, faculty members, research scholars, and technical staffs for their wholehearted support beyond their comfort zone. Once again, I renew my gratitude to all those who back this occasion with a sense of fulfillment. Thank you all. Have a great day.
Thank you.
Thank you, participants. Again, we will rejoin tomorrow at 10:15 a.m. for the next 2 sessions. You can use the same link for access the program tomorrow. Thank you very much.